Okay. Right here, the electron transport chain take place in the inner membrane of the mitochondria. So the folding increase the surface area. Let's see what is this oxidation phosphorylation later. So NADH, FADH product produced in the early stage of aerobic respiration are oxidized by transferring electrons through electron transport chain and finally to molecule of oxygen. The electron transport chain is located in the membrane and composed of series of protein and non-protein molecules. And they are important for the movement of electrons and protons across crystal. Okay, now they here use very minimum amount of text. It's very confusing in this resource book what it says. Uh, Electrons, movement of electron and proton across crystal. So what does it mean? Movement of electrons and protons across crystal. It has a very bad meaning. Both electrons and the protons of hydrogen ion, same hydrogen ion and proton are the same thing across crystal. So crystal is this across the crystal. So it could be we can understand just like a movement like this, across the membrane. According to the sentence, it can cross the membrane. Now actually it's two different things. So here coming back, NADH back into original versions because enzyme catalyze the reactions, electron and proton release. FADH, same thing happen. Now, if you check the light dependent reaction, I don't know whether you can remember light dependent reaction. In light dependent reactions, the first step is PS2. When light is absorbed by the PS2, electrons are emitted by chlorophylls. Electrons are excited by the chlorophylls. Then the electron release, this electron moving down to protein complexes to PS1. And if you check, we have learned the energy of the electron is used to pump hydrogen ion in. You can check it, right? You can check it. This electron energy is used to pump hydrogen ion in. Now that is, it says across the membrane, the hydrogen ion move in. Right? So I don't have to show it now, the notes. So PS2, hydrogen ion pumps in. But here what's going to happen, this proton or hydrogen ion pump out like this. The complexes, the protein complexes act as the proteins transport protein and it transport hydrogen ion out. Same with this, hydrogen ion pump out. Now the proton is accumulate in the inner mitochondrial space. Between outer membrane and inner membrane, this proton build up. Okay, now we can understand NADH and FADH coming with a lot of energy. This energy taken from glycolysis. This energy is in the form of electron and protons. In electron transport chain, they come back to the normal form again, NADH and FADH, and the electron and hydrogen are released. Energy released. Electron moving through these complexes, one, two, three, four, and then complex 4, electrons move out, exit, and this electron has no energy at the end. So if I show you this electron, yellow color, circle around it, and this electron has a yellow color circle around it, it shows the energy of this electron. But when it comes to end, there is no energy. Energy is used to pump hydrogen ion out. The energy is used to pump hydrogen ion out. So they don't have any more energy now. Okay, then these electrons cannot exist like this because electrons need some atoms to move in. So what is happening at the end? This electron combined with the proton, hydrogen ion, and very important thing coming up now, oxygen, to make water molecule. So oxygen is the electron acceptor. So we call it a final electron acceptor is oxygen. That's very important. Final electron acceptor is oxygen. That's why we have to breathe. That's why we have to take oxygen always. Because whatever we take from the nose, the oxygen molecules going to the end like this, and it forms water molecules. Without oxygen, the electron cannot uh, form water molecules. Then no more energy produced. Respiratory chain stop. Aerobic respiration stop. So if we don't provide oxygen, cell stop respiration because oxygen is needed at the end to accept electrons. Okay, now you understand, just similar to the photosynthesis, electron need a place to stay peacefully. In light dependent reaction, electrons of PS2 going to PS1. PS1 electron is going to NADPH. NADPH. In electron transport, electron is accepted by oxygen to make water molecule. Okay. The last, what is going to happen? Therefore, the molecule oxygen is the final electron acceptor in the aerobic respiration in the electron transport chain. And unfortunately, many things are missing. ATP synthesized by oxidative phosphorylation. And further, again missing in this electron transport chain, energy is released progressively from NADH and FADH, and that energy is synthesized ATP. That's it. But it's very bad. Photosynthesis, we learn everything, but electron transport chain, there are so many things are missing. ATP is formed. Now, if I say in this reaction ATP are formed, you lost because you don't know where ATP are formed and you have no idea. Okay, you can do one thing. If you remember light dependent reaction, it is very similar thing happen here. Only small difference. The difference is like this. One difference is look at light dependent reaction, hydrogen ion pump in, but here hydrogen ion pump out. That is one difference. Then light dependent reaction, hydrogen ion accumulate inside the thylakoid. Hydrogen ion accumulate inside the thylakoid. Here hydrogen ion accumulate in a intermitochondrial space here between these two membranes. Hydrogen ion accumulate. If you look at light dependent reaction, inside thylakoid so much of protons or hydrogen ion accumulate, it make the gradients. One side is low, other side is high. That is the gradient. In this case, now the matrix, this area is the matrix, low hydrogen ion, intermembrane space, high hydrogen ion. Then hydrogen ion, in light dependent reaction, we learn hydrogen ion can move now inside thylakoid to the stroma. Just I draw it here again. I mean, you learn it, but I know that you can't remember everything. But I recall quickly that's a thylakoid membrane because chloroplast is like this and the thylakoids are like this. And I'm drawing here the thylakoid membrane. This is the stroma. What I said, this is a PS2. This is a PS1. And I said light is absorbed by the PS2 pigments, chlorophyll, and carotenoid-like pigments. Then in the reaction center, primary electron acceptor is here, and the chlorophyll A molecule release electrons. So then the electrons release out from the chlorophyll A molecules. And these electrons moving through these complexes like this. Then this energy is used to pump hydrogen ion in because this side is a stroma, this is inside thylakoid. Then hydrogen ion pump in. Then it make inside hydrogen ion high, outside low. PS2 light absorb, chlorophyll A release electrons, and this electron is moving through the complexes, and this electron join with NADP plus H plus electron to make NADPH. And lost electron from PS1 is replaced by the electron from PS2. This electron is coming to PS1. And then we learn at last this hydrogen ion has a lot of potentials, and this hydrogen ion can move only with this enzyme called ATP synthase. When it moves out, this energy is used to make ATP. So this is the summary of a light dependent reaction again. We have learned this, and this is again summary of light dependent reaction. Okay, what is important now from this light dependent reaction to us? There is a hydrogen ion gradient created, high, and this side is low. And the energy of the electrons used to pump hydrogen ion in. This energy cannot destroyed and again trapped in the ATP molecule when hydrogen ion is moving out. Now you can understand the energies of the electrons finally move to the ATP. So this energy is coming from the sunlight. Sunlight energy move to the electrons, finally it goes to the ATP. Okay, coming back again, electron transport chain, similar thing happen. Different is hydrogen pump from matrix to inner mitochondrial membrane, intermitochondrial space, right? This is the intermitochondrial space. Now this side hydrogen ion level is high and inside hydrogen ion is less. So this is the potential gradient, same as photosynthesis. Now hydrogen ion can only move back through this ATP synthase complex. This is the ATP synthase complex. Same as light dependent reaction, this electron is moving in, sorry, the hydrogen, right, not electron. Then ADP plus phosphate produce ATP. Now this way of production of ATP is called oxidative phosphorylation. So this method is called oxidative phosphorylation. So it is actually a coupled with electron transport chain. It is not a single reaction like in substrate level phosphorylation. Substrate level phosphorylation, A convert to B, energy release, make ATP. But here complex process, it's a series of reaction, so we call it oxidative phosphorylation. In this way, ATP is produced inside the mitochondria. So I think the confusion in the resource book, ATP is synthesized by oxidative phosphorylation. You can remember like that, ATP is synthesized by oxidative phosphorylation. But since you know a light dependent reaction, it's a little bit slightly different thing happen here. Just like a light dependent reaction, sunlight is used. We call photophosphorylation. Photophosphorylation. Here we call oxidative phosphorylation because oxygen is involved, and it's a series of reaction like this. For better understanding, we need to learn a little bit more than the resource book, only for understanding purpose. Okay. Do you have any questions about the electron transport chain and the oxidative phosphorylation? This is photosynthesis, right? Finally, we need to calculate the ATP level. What is the total ATP level, right? So we do like this. Uh, we just make a small note like this. Substrate level, phosphorylation. So we have seen glycolysis, net production is 2, Krebs cycle, 2 ATP. Rest all formed by oxidative phosphorylation. That means NADH, FADH make ATP by electron transport chain, that is called oxidative phosphorylation. So let's see how many coming now for this place. Glycolysis, there are 2 NADH here, products 2 NADH from glycolysis. So I take this 2 NADH here. Link reaction, 2 NADH. Krebs cycle, 6 NADH. Okay, then FADH is only coming from Krebs cycle 2 FADH, right? 2 FADH. Okay. The theoretical calculation is 1 NADH molecule can make 2.5 ATP molecule. Can make 2.5 ATP molecules. So then we have to multiply by 2.5. Why? We need this conversion. 1 NADH can make 2.5 ATP. This is done by the methods by scientists, and they come up, okay, this is the conversion rate NADH to ATP. And here 2.5 and here 2.5. Then FADH is different. 1 FADH can make 1.5 ATP molecule. So this should be 1.5 then. So let's calculate this. This should be 5. This should be 5. 2.5 multiplied by 6, 15. Then 2 multiplied by 1.5, 3. So I add all now. In all the steps, ATP number I can add now. 15 plus 5, 20, 25, 28. So oxidative phosphorylation, total number of ATP is 28. And substrate level phosphorylation, total number is 4, right? I think we need to remember substrate level 4, oxidative phosphorylation 28, right? And in case for the same side, we better to know what is the ATP production from Krebs cycle. Like this also, I mean, we can calculate this, but at the moment, we try to remember at least this substrate level 4, oxidative phosphorylation 28. Then the total is 4 plus 28, 32. That is the total. That is the total ATP production, 32 ATP molecules. Right, there is a small thing to add here. This 32 ATP molecule is not actually the total ATP molecule produced by the cells. This is only possible for cells like heart muscle cells. Heart is made out of muscles. Muscle cells they are called cardiac muscles. And the cells of the liver cells. So they can only make 32. Other cells make 30. So theoretically, it is 30 ATP molecule produced by the cells except heart muscle cells and liver cells. Why is that heart muscle cells and liver cells produce highest? Because their energy demand is high. They need more energy. So there's no chance to waste the energy, no wastage of energy. So they maximize production of ATP. Why other cell lose ATP then? Because it is like this. Now if you take a cell, this is a mitochondria. Can you remember glycolysis, glucose molecule break down into a pyruvate and it produces NADH and also ATP and other molecules. Now this NADH should move into the mitochondria. I think your basic knowledge, you can understand some transport method use energy. Example, active transport. Active transport need energy. So NADH moving to the mitochondria, it use some energy. That's why two ATP molecule cannot use. But liver cell, muscles, cardiac muscle cell, heart muscle cells, they optimize not to waste this energy. So here, in this transport across the membrane, used energy by all other cells except heart muscle and liver muscle. They have a different system, but we don't want to learn what is the system and why they can do this, but just for our understanding, it is the reason for this kind of a energy lost. So ATP production total 32. Only liver cell, muscle cells can make 32, other cells use 30. Substrate level 4, oxidative phosphorylation 28. Uh, Okay, do you have any other questions, anything to ask? Because we have completed now aerobic respiration. Summary, glycolysis takes place in the cytoplasm. It produces pyruvate. Pyruvate enter into mitochondria. Then acetyl-CoA produce. Acetyl-CoA enter into a Krebs cycle. Krebs cycle completely oxidized. And there are two energy productions. One is substrate level. One is oxidative phosphorylation. So after oxidative in oxidative phosphorylation, the NADH and FADH release the energy, and similar to the electron transport, the light dependent reaction method, mitochondria use the same kind of method. So then hydrogen ion pump out, and this energy is used to make ATP by the ATP synthase complex. The final electron acceptor is oxygen. So now you can understand, we learn the aerobic respiration is glucose, oxygen, it gives carbon dioxide, water, and energy. 6, 6. So this was the original reaction we learned at the primary level, but now we know why oxygen is important, where carbon dioxide produced, where is water is produced, and where is the ATP produced, and what happened to the glucose molecule like that. End. Aerobic respiration is end. Any question from your side? If you don't have question, we have a small section to learn, anaerobic respiration.
Sir,
Yes.
so can you show again that calculation part?
Oh, yeah, I know that most of the biology students they have the problem with math calculation, but it's not that difficult maths. Only thing is you need to remember amount of NADH produce at different level, and we have the conversion rate. One NADH can make 2.5 ATP, one FADH can produce 1.5 ATP. So then we multiply always NADH number with 2.5, 2.5, 2.5, FADH 1.5. So this is the production of molecules during this aerobic respiration. Then we get the each level total number of ATP 5, 5, 15, and 3, 28. The glycolysis 2, Krebs cycle 2 ATP as substrate level. Substrate level 4, oxidative 28. So then 32 is the total ATP production by adding substrate level and oxidative phosphorylation. Yes, hard thing is to remember this kind of information. I know that is a difficult thing for almost all students for such complicated processes like this. I will send this document as well, so you can keep this with you, and maybe use can as a short note with the detailed notes. Can check it again. Then be okay. I think when you see something first time, seems to be complicated. So if you maybe I don't know whether you have experience. I think not. You don't have yet. You know, when we're going to learn the driving, the first step is very difficult. The guy is sitting here. Okay, now you have to check this mirror, that mirror, this mirror, and check, put the signals. Then you lost which step I should do now, which step I should do. Like this, so many things on your head. You feel already almost that this is impossible because there are 10, 12 sets of information you have to check very quickly before you put in the car onto the road. But the day you are confident, you don't try to remember the steps. It's like a naturally coming up. It's you are not thinking I should do this. It's just coming naturally. I think learning is also sometimes like this. It seems to be very complicated and so much of difficult. But good thing is the capacity of the human brain. Uh, three, can't remember, can't remember anymore. Capacity 3 gigabyte, 3 billions gigabyte. 2.5 million petabytes. It's like a something to understand. To put that to perspective, if your brain worked like a DVD, then you would have to leave the television running for 300 years continuously to fill 300 2.5 million petabytes. So we can run a DVD 300 years continuously, that much of data we can store. So it's a good news. We have so much of capacity. We can use that capacity. Initially, it seems to be very complicated, but it is not that complicated. Another thing I should say uh, that you want to understand, you are not doing difficult thing. When you're doing O level and when you're doing A level, the gap is like this. And you think O level is the hardest exam in your life. Once you've done your